Yeah. So here, antiprotozoal drugs, these are the classes of the antiprotozoal drugs. Okay. And malarials, which we discussed, and amoebiasis that we are going to discuss now. And caused by this infection is caused by protozoal organism, that is Entamoeba histolytica, and other drugs that is used for the parasitic infection like pneumocystosis and toxoplasmosis, leishmaniasis, and trypanosomiasis. So these are other protozoal infections. And these infections particularly we are not going to discuss in detail. And this is the infection amoebiasis, which we are going to discuss in detail. So we know that these infections, that is protozoal infections, are caused by protozoa organism, okay, and which are of single-celled animals. And coming to the amoebiasis also you can call it as amoebic dysentery, caused by the protozoal organism, that is Entamoeba histolytica. This is how the Entamoeba histolytica looks like if we go for the high resolution image of Entamoeba histolytica. And it looks, this is the shape and how it looks like. And what are the symptoms of amoebic dysentery, okay, or amoebiasis? So the symptoms generally include abdominal pain or diarrhea and bloody diarrhea. And then these are the main symptoms. If you have a amoebic dysentery or amoebiasis, which is caused by Entamoeba histolytica, you will have abdominal pain, diarrhea, bloody diarrhea. So that means you find the blood in your stools. And next, complications, coming to the complications. So inflammation and ulceration of colon, okay, and with tissue death or perforation, that is the tissue death, necrosis, and which may result in peritonitis, okay. And then sometimes people may have the anemia because you have the loss of stool, that is blood in stool, so you will have the anemia also. These are the symptoms. And then complications, you will have the inflammation and ulceration of colon, that is main, and tissue death and formation of abscess, that is abscess means where you will have the, in abscess, that is a place where you have the bacteria and pus or dead cells, that we call it as an abscess. Formation of pus, we call it as a abscess, okay. So that occurs with the abscess, occurs with the tissue death, okay. So the complications, inflammation of and ulceration of colon, and with tissue death, and this tissue death causes formation of abscess. Abscess means where you have the pus with, that is cell death, the dead cells. And then you will have the anemia. And the sorry sorry the amoebiasis, it is caused by, you know that it is caused by protozoal organism, that is Entamoeba histolytica, and it exists in two forms. One is trophozoite form, another one is cyst form. And trophozoite form is not an infective form, it cannot cause the infection, okay. It is only invasive organism, it can invade the organs, okay. And it can go to the intestine, from intestine it can reach the liver, and from liver it can reach the lungs, or from, okay, and from liver it can reach the heart, or from liver it can, from heart it can reach to the brain. So, and here this is invasive form. And then this is immature form, you can call also, which cannot cause the infection. That means it cannot cause the cell death, as I told you, complication. So in the complication, the Entamoeba histolytica, it can cause inflammation, ulceration or colon, or tissue death, or tissue that I called you, we already told you that it is a formation of abscess. And here if you see that trophozoite, it only can migrate from one organ to another organ, and it cannot cause the infection. That means it cannot cause the cell death or ulceration. And then this is the next form is the cyst. Cyst is the mature form of, okay, Entamoeba. It is infective form, okay. So it can cause the colon ulceration or liver abscess, okay. All these infections can be caused by the cyst form, okay. So these are the forms of Entamoeba histolytica: trophozoite form and cyst form. This is trophozoite form is invasive form, and whereas cyst form is infective form, mature form, and immature form, and it is a quadrinucleic. Okay, you can see here it is mononucleic, and whereas it is quadrinucleic, 4 nucleus you can see here. And this cyst form can further divide into 8 cysts or 8 trophozoites. Okay, this cyst form can further divide into 8 trophozoites. Okay, next, coming to the here life cycle of this Entamoeba histolytica. So this Entamoeba is like a transmission is to the contaminated water or contaminated food. Once if it is ingested, okay, and that is in the cyst form, it will be injected. Okay, so here the transmission of Entamoeba histolytica to the host or we the humans. Okay, so it is happens to the ingestion of or taking contaminated food or water. Once you ingest this, then in the food you have the mature cyst. Okay, and this mature cyst undergoes the excistation, that means multiplication, and then you have the trophozoite. Cyst will be converted into trophozoites. Okay, and this trophozoites will go to the, that is will be present in the small intestine, and then they migrate to the large, that is large intestine or large intestine, from small intestine to large intestine. So here the life cycle, if you see, only 2 phases will be there, and this entirely happens in the human itself or the host itself. So you ingest, okay, contaminated food or water, and this contaminated food or water will have the cyst, and cyst will be converted into trophozoites. Okay, that is converting cyst into that is trophozoites. We call it as encystation. Encystation is that cyst into trophozoites. Okay, infective to invasive. And then, so this happens, all this happens in the large intestine, and then they move to the colon. Okay, and then there what happens is that the trophozoites, then they undergo the binary fission. Okay, these trophozoites can undergo the colon, and then from colon to large intestine they move. Okay, so here the trophozoites will be present in the large intestine. There may they undergo the binary fission, and some of the cysts, okay, some of the trophozoites, okay, so they will be released off, and then they will be converted into mature cyst forms, and or some will remain in the humans, okay, host, and then they undergo the maturation. That means further trophozoite will be converted into cyst form. So trophozoites will be released, okay, through the, that is fecal matter or stools, and some of the trophozoites again they will be further converted into, that is cyst forms, and here the trophozoites can become invasive. Okay, so they can go to the lungs, and then they can go to the liver. Okay, and where they, from there they can go to the brain also. They can cause them that is amoebiasis in brain. They can cause the amoebiasis in the lungs. They can cause the amoebiasis in liver. So the trophozoites can invade. Okay, so the first step is here, if you see the life cycle, mature cyst, that will be converted into trophozoites, then they undergo the binary fission, and you have the here, okay, trophozoites which can invade other organs, and or the trophozoites which will be released, okay, to the stools, and then they get converted into mature cysts. Okay, so this is the life cycle. So the invasion can be, that is from intestine to the liver, and from liver to lungs, and the liver to heart, or that is heart or liver to brain. So this invasion happens, and once invasion happens, then the trophozoite will be converted into mature cyst. Mature cyst, this mature cyst only can cause the infection. These all forms are invasion. They can invade to different organs. Okay, so that is the life cycle of your Entamoeba histolytica. Then coming to the pathogenesis. So as I told you already, so the pathogenesis, how the life cycle will be there in your host, so that is ingestion of cyst. Okay, and then the cysts will be through excitation process in the small intestine. You will have the production of trophozoites. Okay, and these trophozoites, so they can undergo the multiplication and colonization in the large intestine, as I already explained you. And then from their trophozoites can invade the other tissues and then cause. Okay, so the invasion can cause the ulcerations. Okay, or excistation, that is encystation. Encystation means they will come out of the stools, encystation, and then further, okay, they can infect the non-infected persons or anything, or they migrate to the blood and then they go to the liver. From liver, so then they can go to heart, or from the heart it can invade. This is trophozoites only, not the cyst. They can invade the brain. So this is how the pathogenesis of amoebiasis happens. Amoebiasis means generally we consider that is dysentery. Okay, and where watery stools or bloody stools. Okay, watery stools are amoebic dysentery we consider, but amoebiasis also can occur in liver. That we call if it occurs, amoebiasis occurs if in liver, we call it as a liver abscess, or amoebiasis it can occur in lungs, or it can maybe in serious cases it can occur in uh, liver and lungs and heart, or it can also occur in spleen, or it can also occur in brain. Okay, so it is not amoebiasis means not only dysentery, that is watery stools or bloody stools. So this is how the pathogenesis occurs. Okay, amoebiasis. Yeah, the difference between cyst and trophozoite, if you see that, as I already explained you, what is the difference between, if it is a mononucleated, okay, trophozoite is a mononucleated, and here it is that you can see that it is a quadrinucleated, and size also you can see. And as a other one also I told you that it is it is invasive form only, and it cannot cause the infection. And other one is that it is infective form, and this is invasive form. That is the main difference. Okay, so we can see the nuclei here. It is a mononucleated, and then this is a quadrinucleated and this one trophozoite, again in between there is a form that I have not shown you here. That is a pre-cyst form. Okay, so trophozoite, pre-cyst, and cyst form. Okay, that's why we have. So the major difference, if you see, invasive form and non-invasive form. Morphologically, see, if you see that it is a mononucleated and here immature form, and you can see that here it is a, for the, if you see that is a quadri, 1 2 3 4 nuclei you have. Okay, so quadrinucleated. So that mature form will be matured. Okay, trophozoite, and it will be converted into cyst form. Okay, so cyst form only is the infective form. Okay, and whereas trophozoite form is invasive form. It can only invade from one organ to another organ. It cannot cause the infection. So to cause the infection, the trophozoite form has to be converted into cyst form, and once if it is converted into cyst form, then this cyst form can cause cell death. Okay, wherever it is invaded, it can cause the cell death, that is through the leakage of ions. Okay, leakage of ions from the infected cell. So only cyst can cause the infection. So then pathogenicity, that in the pathogenicity, the mode of transmission, we know that that is a mode of transmission, ingestion of contaminated food or water, and route of transmission, that is fecal oral route majorly. So since we know that cysts will be excreted through the fecal route, and then it is transmitted through the oral route. Okay, we take the food, and this food or water ingested through the oral route. Okay, so either contaminated food or water, source of the infection. So here flies, house flies, they can transmit, that is house flies, generally cockroaches. And mainly if you don't maintain the sanitation, that is hygiene, okay, and poor sanitation, if you have, all these things can happen. Okay, that is infection can occur. And then under the pathogenicity you have the amoebius, types of amoebiasis, that is intestinal amoebiasis. Already we discussed that intestinal amoebiasis. You have the dysentery, that is watery stools and then bloody stools. And bloody stools happens if the cyst form causes the cell death. Okay, cell death, that is where it is infected. Then you have the bloody stools. So, and what is 1 to 4 weeks? That is, it can be symptomatic or asymptomatic. This is the intestinal amoebiasis. Okay, and next one is that liver amoebiasis, that is extraintestinal, that is, or you can call it as a hepatic amoebiasis. And then you can see that here hepatic abscess formation. Okay, abscess formation where cell death is there, cell death in the liver. Okay, and transmits through the portal veins from intestine to liver. The transmission is from intestine to liver, that is invasive form. First, that is invasive form will be there in the intestine, in the large intestine, and from there it can transmit, or okay, through the portal veins to the liver, where invasive form will be converted into cyst form and forms the liver abscess. Liver abscess. And pulmonary and amoebiasis, and the pulmonary amoebiasis you can have, that is transmitted from liver to pulmonary lesions. Okay, that is transmission is from liver to the lungs. And then cerebral amoebiasis, and here cerebral amoebiasis is from liver to heart. Okay, and then from heart then to brain. Okay, so that is what happens. So amoebiasis, it can occur, as I told you, liver, heart, and then brain, cerebral amoebiasis. So this, you can see that first formation here, cerebral amoebiasis, the yellow colored, you can see that first formation, that is because of the death of brain cells. Okay, and that form, you can see that. And the drugs, okay, what are the drugs we use, okay, for the treatment of amoebiasis? So the drugs which are used for the treatment of amoebiasis are and that is the treatment of either dysentery or bloody stools. We call them anti-amoebic drugs. And the drugs are, the classification is that it is not based on the chemical, there is not a chemical classification or based on the chemical features of the drugs. This is where we target, what we target. Okay, whether it is systemic amoebiasis, systemic means except intestinal. Okay, either it is systemic or intestinal. So in the intestinal amoebiasis, you can be luminal, so which can okay, so luminal, under that first one you have the luminal, so active only against the intestinal forms of these luminal anti-amoebics. So luminal anti-amoebics, they are active against only the intestinal forms of amoebia. So they cannot, you cannot give them to treat the systemic amoebiasis. That is, if amoebiasis occurs in lungs, or if amoebiasis occurs in brain, or if amoebiasis occurs in spleen, or if amoebiasis occurs in heart, you cannot treat those amoebiasis by using these drugs. These drugs only for the intestinal amoebiasis. So example you have are the diloxanide, idoquinol, and pleoquinol. So these are the drugs which are used for the treatment of luminal amoebiasis. Okay, the first class. So if you want, you take down these examples, write down these examples. Once you finish writing down these examples, you just raise your hand. So diloxanide is diloxanate furoate. Okay, this is furoic acid is there, furoic acid, and it is a prodrug. Okay, and this is a prodrug, diloxanate. It is not diloxanate, diloxanate furoate. So furoic acid, furan, and it is carboxylic acid group, furoic acid attached, and it undergoes the Okay, here hydrolysis, and you'll have the diloxanide. And diloxanide, if you see that, it chemically resembles chloramphenicol. Okay, chemically it resembles chloramphenicol, diloxanide. And for all these drugs, okay, diloxanide and cleoquinol and idoquinol or whatever these drugs, the exact mechanism has not yet been proposed, okay, are not yet known. This exact mechanism, how they act on the Entamoeba histolytica, that is protozoa, amoeba, amoebiasis, but till not there they have proposed that they may inhibit the DNA replication, RNA processing. So these only proposed mechanism of actions we have, but exact mechanism of action, how they kill this Entamoeba histolytica, is not yet been known. So here they say that since it is looks like chloramphenicol, okay, and then it will may follow the path of chloramphenicol mechanism of action. And here these are quinoline derivatives. So they are also not exactly, mechanism is not. Only the proposed mechanisms are they may inhibit the DNA synthesis or they may inhibit the RNA synthesis. So these are only the proposed mechanisms, but the exact mechanism is not yet known. So once if you write, and you can please raise your hand so that I can go to the next slide. Okay, so I go to the next slide. And luminal trophozoite. Okay, so here these drugs, okay, so again they are for luminal, that is intestinal infections, and examples have metronidazole and then tinidazole or ornidazole. Okay, ornidazole, you have tinidazole and ornidazole. These are against only trophozoites. Okay, trophozoitocidal, these are luminal trophozoitocidal, and they will not act again on the cyst forms. So these act only trophozoites, and example you have metronidazole, tinidazole and ornidazole. Okay, this is the second class of the drugs. So, and these drugs active against the intestinal forms. So we know that intestinal forms can be cyst form or can be of trophozoites. So here these drugs are only active against the trophozoites, not on the cyst forms. That means they are active against the invasive forms, not active against the infective forms of Entamoeba. So example, metronidazole, tinidazole and ornidazole. So you write down these structures, and for all these common is these are imidazole. Okay, you have the imidazole, and this to nitromethyl imidazole, and but this alcohol portion in these two cases, all three cases, the alcohol portion changes. So methyl nitro imidazole, okay, 2 methyl, this is 1 2 3 4 5, 2 methyl, okay, 5 nitro imidazole derivatives. All will have common is that you have the 2 methyl 5 nitro imidazole, and only the side chain attached to the nitrogen of the imide, first nitrogen, will change. Okay, so then in this way it is easy for you to remember. So once you write down the structures, and do raise your hand so that I can proceed to your next slide. So okay, I'll move to the next slide. Okay, coming to the next slide is, yeah, coming to the mechanism of action. So the mechanism of action of metronidazole, this is how the mechanism of action metronidazole, and we know that this is the metabolic pathway of amoeba. Okay, in the metabolic pathway, we know that here in this, what happens is that metabolic pathway here pyruvate will be converted into acetyl-CoA, and this is coenzyme A, and coenzyme A will be converted, will combine with the pyruvate, and then pyruvate to acetyl-CoA, that is coenzyme A, acetyl coenzyme A. This is the metabolic pathway. So this metabolic pathway is for the generation of energy. Okay, so we know that we have studied in humans, okay, how the generation of energy will take place through the glucose metabolism. Okay, and then once if it is glucose, then the mitochondria through the electron transport chain, and how the ATPs are generated. It is similar to that. Okay, it is electron transport. So through the reactions, okay, oxidation and reduction reactions, the electrons will be transferred to the oxygen, and once the electrons are transferred to the and generation of oxygen is there, and this oxygen finally will be utilized to generate the energy, that is ATPs. So that is oxidative phosphorylation. Same way, the entamoeba also have the, okay, has the metabolic reactions. Of the metabolic reactions, if you see that here, pyruvate to acetyl-CoA, this reaction is done by pyridoxine. Okay, this is pyridoxine oxidoreductase. Pyridoxine oxidoreductase. This is the enzyme which converts pyruvate to. This is not there in the humans. This is only there in the entamoeba. Okay, so this enzyme pyridoxine oxidoreductase helps in formation of acetyl-CoA, and oxidized form. Okay, Elbic pyridoxine oxidized form will be converted into pyridoxine reduced form by converting this into this. So pyruvate plus coenzyme A will be converted into acetyl-CoA or acetyl coenzyme A by the enzyme pyruvate pyridoxine oxidoreductase. Okay, and during that, oxidized form will be converted into reduced form, and then normal metabolism I am telling you. Okay, this is normally what happens. And pyridoxine again, it will be converted into oxidized form, reduced form. Further, it will be converted into oxidized form, and by transferring the electrons to NAD, okay, H to NAD plus, okay, NAD plus to NADH, and then again, finally, electrons will be transferred, and you'll have the oxygen that is being converted into water. So this is normally what happens. Okay, now what we are giving is that, okay, metronidazole. If you see that metronidazole, it is having a nitro group. Okay, so this nitro group, okay, tropozoid crosses through the trophozoite membrane, and then this reduced form is the pyridoxine reduced form. Okay, converts this nitro form into a radical form. Okay, radical form, and this radical form releases superoxide. Okay, and here don't write it down that it is, okay, superoxide will be released. Okay, so this, whatever the things till now I explained you, this is normal metabolism. So in the normal metabolism, what happens is that this pyruvate ferroxidine oxidoreductase converts pyruvate to acetyl-CoA, and then from there electrons will be transferred through the oxidation and reduction, and oxygen will be generated, and this further it will be used for the generation of energy. But when you give metronidazole, so this metronidazole is having a nitro group. I told you that is 2-methyl-5-nitroimidazole. This nitro group, okay, it crosses the membrane, and then you have here reduced, okay, pyridoxine. This reduced pyridoxine, what it does is that, okay, to undergo the oxidation, okay, so it converts nitro group into superoxide, a radical one. Okay, and then you are not, don't be confused with this. This is superoxide dismutase. So this is only present in the Entamoeba histolytica, superoxide dismutase, that you don't know, don't worry about that. So here nitro group will be converted into that is oxygen, that is which is having a radical one. Okay, so this radical, okay, this radical, that is superoxide, okay, this radical, or which gives the radical oxygen, so which gives the superoxide, and from this superoxides will be formed, and these superoxides we call them as a reactive oxygen species. RNO2 nitro group is converted into nitro radical, and this radical gives you superoxide, and these superoxides are called as a reactive species, and these reactive species are highly reactive and can damage the DNA. Okay, they can damage DNA. So you have to understand that metronidazole nitro group will be converted into radical form, and this radical form, okay, gives you superoxide, and these superoxides are very reactive, highly reactive species, so they can damage the DNA. How they damage the DNA? Okay, how the damage the DNA? So nitro, see, you can see that here. Okay, here you can see that metronidazole. So the metronidazole, what it does is that, okay, so pyridoxine, that is reduced form will be there. That reduced form, it converts metronidazole nitro group into a radical group, and from that superoxide will be released. Superoxide will be released, and this superoxide, it is very highly reactive. So you can see the reactive oxygen species. This is a O2 oxygen diatomic, and then this is hydrogen peroxide. You can see the red dot, that is free radicals, and then this is a superoxide. You have only one lone pair of electron, and this is hydroxy radical. So these all, whichever the oxygen species, whichever having lone pair of electrons, they are called as a super, okay, super, that is reactive oxygen species. These reactive oxygen species are produced, okay, from the metronidazole, that is by peroxidine, okay, peroxidine, and these reactive species can damage the DNA. Okay, how they damage the DNA? So here you can see this is thiamine. So thiamine is converted into thiamine glycol, or this is guanine. Guanine is converted into, okay, guanine is converted in, here you can see that hydroxyguanine. So, and here these are all, okay, can block the replication. These are false metabolites. Okay, thiamine is actual metabolite, and this is a actual metabolite. These are all false metabolites. These metabolites can stop the DNA. Okay, and here also you can see this is also conversion of guanine into 8-hydroxyguanine, which is mutagenic, because, okay, so mispairs with the adenine. So in this way, they can cause the DNA damage, and thereby it prevents the cell replication and finally cell death. This is the mechanism of action of metronidazole or imidazole derivative. So imidazole derivative, so they release the superoxides, and these superoxides or can cause the damage of proteins, also can cause the damage of DNA also. If there is a DNA damage and cell cannot replicate, or it cannot produce the enzymes which are required for it. Okay, so this is about the mechanism of action, and if you have any doubt, okay, so you can ask me. So if you have any, I unmuted you, and if you have any doubts, you can ask me. Are there any doubts? Okay.